Selection of traits in organisms has been used since the invention of agriculture by man. The Bible excerpt describes a man who artificially selects animals for their strength. It seems a simple concept to breed your animals so they have more favorable traits. This concept has also been applied to plants. 10,000 years of classical breeding has made plants significantly different from their ancestors. One example is Brassica oleracea. From this one species, Brussels sprouts, cabbage, and broccoli have all been bred. Broccoli, B. oleracea italica, is thought to have diverged from its close relative cabbage by the Etruscan civilization, who flourished in Tuscany during the 8th century to 3rd century BC.
This practice of selective breeding is thought to have been an unintentional process during the Neolithic Revolution and domestication of plants, when hunter-gatherers began to settle down and start planting seeds near their home. Now, horticulturalists have this practice down to a science, knowing how to isolate certain genes through the breeding method.
In animals, a similar process has occurred over the centuries. The major difference from plant breeding is that there is a much longer time between generations. A prominent example is the species Canis lupus familiaris, or the domesticated dog. It is estimated that dogs began to deviate from their close relative Canis lupus lupus, the gray wolf, about 12,000 years ago during the beginnings of agriculture, when dogs were being isolated for different uses. Hundreds of unique dog breeds now exist. Humans have even taken this idea of breeding for desirable characteristics to their own species. Eugenics, by medical definition, is the applied science or the biosocial movement which advocates the use of practices aimed at improving the genetic composition of the population. Eugenics has been used by many social groups as a method of power exhibition. In the United States, people with mental disorders like schizophrenia, bipolar, and depression would be sterilized because scientists believe that there was a hereditary nature to some diseases. Many states passed laws requiring sterilization to be done on all mental institution patients, such as the state Indiana. The Carnegie Institute suggested euthanasia as the best way to cleanse a population. People without Anglo-Saxon heritage were considered by many eugenics organizations as inferior. The Immigration Restriction League lobbied for an immigrant literacy test to prove their inferiority. Targeted groups included Hispanics, Southern Europeans, Africans, and Native Americans. Many of the American eugenics policies rubbed off on Nazi Germany, who used these tactics against several ethnic groups in Europe, most namely Jews and Gypsies. The practice of breeding has been a system of trial and error for centuries, but new technologies have come onto the radar of scientists of multiple fields. Techniques that cut out the waiting time of several generations which classical breeding requires. It is collectively referred to as genetic engineering. Genetic engineering seems simple enough. Just cut and paste a gene from one species to another. Let's begin with a quick explanation of the process. DNA is agreed by biologists as the genetic material. Chemically speaking, a clean break in the DNA sequence which determines a trait first requires the knowledge of the location of genes on the chromosome which holds that sequence. Expensive proteins called restriction enzymes help make the cut into the DNA at both the donor and receiver chromosome. The fragment of DNA will then be extracted and stimulated at the receiver chromosome to bond with it, resulting in a hybrid genome. Not only is it expensive to acquire restriction enzymes, but it is also time-consuming to determine whether to make the chemical cut. This system was first used by the team of Herbert Boyer and Stanley Cohen, who in 1972 completed the first genetic modification with a bacterial plasmid and auxiliary chromosome to the bacteria. Boyer had isolated the restriction enzyme EcoR1. Cohen had developed methods to both clone bacterial plasmids and introduce foreign plasmids to bacteria. Their combined efforts led to the development of a huge international industry. The work which this scientific team completed has had an enormous effect on several industries, from breeding to agriculture to medicine. Let's begin with pharmaceutical products. Genetically modified organisms, GMOs for short, have been used to create drugs and medicines. One of the most simple genetic modifications is that of insulin-producing bacteria. Insulin is a hormone which controls blood sugar in humans. The human gene for insulin production is isolated, then put on the bacterial plasmid which is cloned. The plasmids are injected to the bacteria, which begins production of insulin. Millions of diabetics now take insulin produced by bacteria or yeast. Medicine does not limit itself to production of drugs. Pharmacogenomics, the study of an individual body's reaction to drugs based on inheritance, applies the knowledge gained about the human genome. Referencing which genes cause what functions, scientists can study the effect of drugs on a person's genome. It also fulfills the promise of personalized medicine, where different persons' drug prescriptions and combinations are chosen in respect to their genes. The goal of this study is to maximize the efficiency and minimize adverse effects of medicinal drugs. Knowledge of the human genome from the Human Genome Project has given rise to this field. Genetic engineering has been applied to medical treatment of humans in another way, by changing the genome of a diseased person. By changing the chemicals produced by an individual, hereditary and previously incurable diseases such as sickle cell anemia and cystic fibrosis can be treated for. A synthetically produced virus is injected with a strand of DNA which has the right sequence. The virus serves as a vector to get the properly coded gene into the individual, effectively infecting the person with correctly functioning proteins. Although this science seems promising, it has many risks. The virus could come out of dormancy, the gene could produce a tumor, and the body may have a negative response to the foreign object. Researchers hope to make the process of gene therapy a risk-free, cheap process with further development, but it is currently still in its juvenile stages. In plants, genetic engineering has been used to increase agricultural yield in a variety of fashions. Modified genomes can provide resistance to diseases, pesticides, and infestations. Certain species that may not even be in the plant kingdom offer genes which code for beneficial traits. Monsanto, the biggest patent holder of genetically modified organisms in the United States, and producer of the popular herbicide Roundup, began testing drought-resistant corn in March 2012. Company claims it will reduce irrigation costs and allow corn to survive periods of dry weather. A gene referred to as Drought 1 was used to alter the genome. The gene causes the plant to be more conservative of water. Biotechnologists have also managed to genetically modify corn to express a gene which makes the plant secrete a natural insecticide, eliminating the need for pesticide to be sprayed on the crops. The former method, epitomized in GM rapeseed, makes the plant resistant to the negative effects of pesticide chemicals. Plants are perhaps the most well-known category of genetically modified organisms, but scientists are still bridging the uses of genetic engineering to animals. We have seen the use of genetic modification methods in humans with gene therapy, but in all humans it is difficult to find a technique as effective as those used in plants. Lower orders of animals have been used more frequently, most namely fruit flies, which are used by biologists to study patterns of inheritance. In 2010, malaria-resistant mosquitoes were created. The gene inserted to the mosquito's genome affects its gut, making the malaria-causing pathogen incapable of developing and, therefore, spreading. Mice have also been subject to genetic modification. Referred to as lab mice by the general population, they are used to study tissue or cell response to disease. As mammals, the result of these experiments may be useful knowledge in medicine. In China, cows have been modified to produce milk with the same properties as human breast milk. Genetic engineering extends to the quasi-science fiction realm of cloning. The concept of replicating an individual organism seems quite abstract, yet it is in practice today. The first mammal ever to be successfully cloned was Dolly the sheep, a Finn Dorset raised in Scotland. 2 donors gave a cytoplasm and a stem cell nucleus, respectively, which were then electrically charged in order to stimulate cell division. The result was a developing zygote, which was injected into the uterus of a female. Dolly lived for 6 years, dying in 2003 of a virus caused cancer of the lungs, which had been spreading around the flock. Other mammals from cats to camels have then been cloned in the years following Dolly. Genetic engineering methods and techniques are diverse and are on the frontier of science, yet the applications have been the subject of much controversy over the past 40 years since its inception. The parties against the science may be agitated by moral, religious, or superstitious beliefs. Bioethics, the study of ethics brought about by advances in the life science or medicine, are a generally overlooked issue which should be brought more present in society today. The next 15 minutes are designed to improve your understanding of the science which will shape our future. Even the most simple modes of genetic engineering receive controversy. The manufacturers of GMO plants such as Monsanto claim that the benefits of GMOs outweigh any risks or costs. A prime example of this corporate propulsion is a product referred to as golden rice. Developed by Swiss biotechnologists between 1993 and 2003, the rice produces the chemical beta-carotene, which leads to the synthesis of vitamin A in humans. Vitamin A helps prevent blindness, and vitamin A deficiency is a common occurrence in third world countries such as India and Ethiopia. To combat this condition that leads to irreversible blindness, various organizations have released the crops to subsistence farmers to grow. However, golden rice does not achieve its intended goal. Many scientists, including World Health Organization malnutrition expert Francesco Branca, agrees that there may be a negative side effect to the growth of this GM crop. Biodiversity of food crops in poverty-stricken nations may be reduced by making the narrow selection of a crop which solves one malnutrition problem. Stressing the importance of vitamin A may overshadow other nutrients, such as the lack of carbohydrates and fats. Fats also play a role in the creation of vitamin A, showing the necessity of a balanced diet for overall health. Branca and Dr. Shiva both suggest either distribution of supplements or growth of natural, healthy plants in order to broaden the spectrum of target foods. By reducing the biodiversity, one would lower the variety in the gene pool due to interbreeding. This could have effects on population dynamics in reaction to specific environmental conditions, like a drought, since less favorable genes are available. A narrower gene pool in this instance could decimate the population. Roundup Ready is a brand name seed produced by Monsanto and is resistant to the herbicide Roundup. In 2006, environmental scientists brought up a court case over the USDA's approval of the alfalfa variety. They claimed that the USDA did not examine the risk of lower biodiversity from planting the crop. In resulting cases, Monsanto appealed to the U.S. Supreme Court over the required USDA environmental study ordered by a Californian district court. In the first Supreme Court ruling over GM crop, although somewhat unclear, the planting of the alfalfa seeds was halted. Despite negative reactions, in 2011, the USDA approved the crop. Cases like these represent the large lobby that the GMO industry has in the U.S. government. Monsanto has even used U.S. diplomats to lobby the Pope in accepting GM foods over natural foods. Approval of the Pope would increase consumption in over 1 billion Catholics around the world. Remember the drought-resistant corn? Despite Monsanto's claims for it to have superior moisture retention, field tests have indicated that they are no more resistant to dry conditions than corn classically bred for dry areas. It will not combat an actual drought, but rather be able to survive drier climates like Texas. Plant scientist Doug Gurian-Sherman, a former biotech advisor for the FDA, suggests that better farming practices with traditional corn would be the better route to take, since the risk of losing biodiversity is lost. Yet not all concerns are based on corporate morals. A well-known GM food is the fish tomato. An antifreeze gene from an Arctic fish was put in the tomato's genome so it could be frozen for longer trips, lowering the number of rotting tomatoes. Some people suggest a metaphor of frankenfood by combining genes of different species, putting pressure on the basis of genetic modification. EnviroPig, a registered trademark, are a variety of agriculturally produced pigs developed by the University of Guelph in Canada. The pigs digest phosphorus from cereal grains more easily, eliminating the need for supplements in the pig's diet, lowering costs. Also, less phosphorus-containing manure is produced, which can cause a chain of events resulting in extreme algae growth in freshwater bodies. Groups claim that the problem is poor agricultural practices, not the way a pig naturally digests food. Despite disputes over GM food, the most tearing debate is over the use of this technology in human. There is an enormous potential for the science of genetic engineering to be practically applied to humans. Like cloning, the idea of transgenic humans seems far-fetched and still in the realm of the science fiction genre. but Homo sapiens are rapidly approaching the point at which they will be able to alter their being. Physical methods have already been developed, like plastic surgery. Would you spend thousands of dollars on noticeable reconstructive surgery if you already had ideal genes? The premise of these so-called designer babies is that a parent will be able to have a baby with their desired traits, whether it be for disease resistance, gender, appearance, or IQ. The procedure is called in vitro fertilization, literally a test tube baby. This way, the sperm and egg are in a controlled environment and many variables are eliminated. It is also very expensive, costing upwards of $10,000 for the fertilization. The embryo will have a very low chance of living in the uterus as well, requiring many costly procedures to keep the embryo alive. Like in plant cases, there is an issue of biodiversity loss in the human gene pool, but the issue of cost is what most concerns people. Some imagine a future where the wealthy with ideal genes will discriminate and segregate those with what will be disabilities, like susceptibility to disease, unfavorable appearance, or lower intelligence. Interbreeding of the two divisions would be limited, resulting in a new area of eugenics. Vision of a dystopian future could turn off the wealthy to this practice, yet most people realize that the rich do not have much sympathy or respect for the condescendingly termed commoners. One of the prominent issues in politics today is stem cell research. Undifferentiated and undeveloped cells, called stem cells, taken from an aborted embryo and then stimulated to a point where they become the desired cell, whether it be that of a bladder, a heart, a nerve, or skin, it eliminates the need for a donor organ. But the donor fetus has to have compatible genes with the receiver. The process of therapeutic cloning eliminates the need of a donor organ or fetus, and solely requires a donor nucleus and donor egg. The donor nucleus from a functional cell is injected into the egg, a process similar to gene therapy. One potential case would be a burn patient. A skin tissue culture could be developed from a donor with similar skin color to the patient and eventually be reattached to the patient's body. The skin would be genetically identical to that of the donor. Even more controversial than therapeutic cloning is reproductive cloning, the formation of an identical human. In this process, an entire human being is cloned, not solely an organ. The clear issue with this concept is what may happen to individuality. The clone would be used for spare parts, but would the clone be soulless and unhappy after its use is complete? How would the feeling of having a copy affect the original person? Aside from these basic human morals, many religious groups argue that it would be meddling with nature and God's intentions. But what is genetic engineering at its core? Is it playing God? When I asked Dr. Mark Schlesman, professor of biology at Vassar College, the same question, here's what he said.
Genetically modifying human beings.
Human beings, yes.
No, I don't.
And why so?
Well, this gets to be a little bit more maybe of a philosophical question than a straight, biological question.
Mm-hmm.
But my feeling is that as far as the supernatural or God goes, that God wants us to use our big brains to figure out how things work. And so I guess one way I would put it is if God didn't want us to do that, we wouldn't have the capability to do it.
However, many religious groups disagree with Dr. Schlesman. Christians say we are messing with God's creation, especially humans, which were created in God's image. Ted Peters, a Christian theological professor, argues that humans tend to co-create with God, contributing to the evolutionary process. Many religious groups are also opposed to stem cell research because abortion is the medium to obtain stem cells. Cloning also takes away the holiness of the human being. Congress has yet to present any legislation over genetic engineering in humans. In March 2012, 55 congressmen sent a signed letter to the FDA requesting them to label GM foods. But when will other things involving genetic engineering come to the spotlight, and when will questions start to be asked? When will the Supreme Court be handling cases about doctors who help design babies with parents? When will our very humanity come into question? Something called the Malthusian catastrophe may become a reality soon. Thomas Malthus theorized that once population growth outpaced agriculture, there would be a return to subsistence farming like in the days before the Industrial Revolution. In the next 20 years, Growth in genetic engineering may lead to a cure for death. Who would not want to live another 100 years and see Paris? Is having more and more food because of advances in agriculture GM foods going to be a necessity? Our population has been growing exponentially for the past 200 years. In 1900, the world contained 1.5 billion humans. Now it is approaching 7 billion. More plentiful, nutritious foods, which may reach our kitchen tables because of genetic modifications, could increase lifespans. Gene therapy and cloning could help increase that too. The currently reproducing generation could end up living to see their great-grandchildren, or maybe even their great-great-grandchildren. Increasing the population as the death rate drops, resources like water and oil will dry up. Spaces for living and growing will run out, and the very basis of life will be pulled out from under us. Carrying capacity of the earth will be reached. War may be created just to wean out those that are weaker. We may even return to the hunter-gatherer mentality, which took us so long to emerge from. People will die off because of overcrowding, malnutrition, or even contamination of resources. The birth rate will fall below the death rate for the first time in millennia, and an apocalyptic scenario will spur panic. Humanity. Falling. Should we have to worry about the maleffects of the science of genetic engineering? Or can we trust that the coming generations will moderate the practical uses of this technology? It is our species' responsibility to keep ethics in balance with knowledge, and not to jump the gun. If you know how something works with its implications, you are armed with the power to be the change you wish to see. Do not, by any chance, overlook it.